This is screen capture video 12.3 for Unit 12, and I was just talking about the replication fork in the last video. This is something that forms at one end of the DNA molecule, the parent molecule. It unwinds and splits apart, and the two daughter strands are formed along it, and the separation continues all along the parent molecule until eventually there's no parent molecule left, and the two daughter molecules exist half old, half new. Here's an illustration from the text showing the chromosome, and this chromosome is made up of the tightly wound DNA, just scrunched together. And what they're showing here, as you go to the left, they're greatly magnifying it. So eventually you can see how the DNA is coiled, and within the coil you have the double-stranded helix. And they're showing these proteins called histones that the DNA molecule is wrapped around. And I don't think I talked about it a great deal in the last unit. I may have, I don't remember. I do remember saying something about calico cats, but the female cats would have two X chromosomes, and at any given time only one of the X chromosomes is being read, the genes on it, and for the color. And that the histones come into play with the DNA being wound around the histones, and whether the genes are going to be exposed or not. At least that's what I was told when I was in college. But here, as you go further to the left, you can see the double-stranded helix. The rungs would represent the nitrogenous bases. And it's interesting to notice how they go from the electron micrograph, taken through the electron microscope, to this illustration of trying to explain what they're seeing. There are some links, and at the time of this video the links worked. However, what I have noticed over the course of years teaching, that just because there's a link on the internet, especially on YouTube, it doesn't mean that the link will always be good. So you're welcome to click on these links, and they'll take you to YouTube videos. I cannot guarantee that they'll still be good, and if they're not, you're welcome to look up some other YouTube videos or videos elsewhere that help describe the DNA replication. I've greatly simplified it, and if you take a genetics class or a cell biology class or a molecular biology class, you'll talk about this in greater detail. I did have another video here, and it didn't work, and there was a mistake in it, and I can't remember if it's this video link. I may have to go back and take a look at that. But it's something I would discuss in class. So just be careful of this video link here and watch to see that it is accurate. So we get DNA from DNA by what's called DNA replication. It's great for the DNA to be, or for the information to be stored in DNA and to be passed from cell to cell. We talked about mitosis and meiosis. That has to occur. But it's important that that DNA can be used in the cell, that it actually will be read somehow. And the part of the process to do that is you have to move the information from this huge molecule, DNA, to a smaller molecule that can be moved around the cell, and that's RNA, ribonucleic acid. And the process by which we get RNA from DNA is called transcription. Okay, as I talk about DNA versus RNA, it might help to think about these a little bit. DNA, the D stands for deoxyribose, that's the sugar that's found in DNA. RNA, R stands for ribose. The sugars are very similar. Most people wouldn't detect the difference between them, but they're very similar, but yet they do differ, just by an atom. The double-stranded helix is usually how we think of DNA, though it doesn't have to be in a helix, but that's generally how we think of it. RNA, We generally think of it as being single-stranded, but it can be double-stranded. If you study viruses, there are ways of classifying viruses, whether they're DNA viruses or RNA viruses, and if they're RNA viruses, whether it's single-stranded or double-stranded RNA. Same with the DNA viruses, whether double-stranded or single-stranded DNA viruses. But for our purposes, we'll think of it as single-stranded. DNA is a very large molecule. It's the largest molecule we've considered so far. RNA is smaller than DNA. It's just a fraction of the size of the DNA. And the 4 nitrogen spaces found in DNA are C, G, A, and T for thymine. No uracil. RNA contains cytosine, guanine, adenine, and uracil, but no thymine. So if you can see a T, that's DNA. If you see a U, it's RNA. And this is how DNA, or how information flows. It's held in DNA, which is an extremely large molecule. There are links on each strand of the chromosome, DNA, that will provide information, and those are called genes. And from the gene, we can produce a molecule called RNA. And so the information that's in that gene is transferred to this RNA molecule. And then from the RNA, we can use the information in this RNA molecule and translate it into an amino acid code or sequence. We talked about polypeptide chains in the past. And the polypeptide chains can be hundreds of amino acids in length. And that protein then gives us our physical characteristics. So here's just a short section of the DNA molecule. We're showing gene 1, gene 2, and gene 3. Typically, the gene is only found on one of the strands of the chromosome, not both of the strands. But they're showing a portion of one of the strands down here. Interestingly, there's no T in here, but it is DNA. Starts out with the A and goes to different letters. This is transcribed into an RNA molecule. And so for where there's an A, there will be a U. Where there's a C, a G. Where there's a G, a C. And if there's a T up here, you get an A in the RNA. It's just not shown, unfortunately. Eventually, every 3 bases of the RNA molecule, and specifically messenger RNA, will serve as what's called a codon. And that codon will code for a specific amino acid, only one type of amino acid. So this codon, 3-letter codon, would code for this amino acid. This 3-letter codon would code for a different amino acid, and so on. As it turns out, there are 3 U's here, 3 U's here, so you get the same amino acid. There is some redundancy in the code, so that different 3-letter codons can code for the same amino acid. It's just not shown here. There are over 60 different codons and only 20 different amino acids found in most organisms. And so these amino acids are joined together eventually in translation and protein synthesis to form a polypeptide. Here we have the different 3-letter codons of messenger RNA, and if you were to count these up, there'd be over 60 of them, and it goes along with this. Say for AUG, this would be a start codon. This starts the process, and of course the AAG codes for lysine, which is shown here, and then they're kind of skipping to the stop codon. They're not showing this 3rd one, unfortunately, but they're showing the phenylalanine here, the UAG. Let me just double check here. UUU. I'm sorry, they are showing it here. The UUU, the triple U, would code for phenylalanine, and then the UAG would be the stop codon. They're just not showing the stop codon down here. In producing RNA, the gene on the DNA molecule has to be exposed so it can be read. So what happens is at the beginning of the gene, the DNA molecule unwinds and separates. But it doesn't do that all along the gene, and especially not all along the DNA molecule. That would not be safe for it. So what happens, the bases are exposed, and actually here is the gene part in the darker blue. These bases are exposed, they attract RNA bases, they come in and attach, and they're bonded together, and this process moves in one direction down the gene. And so the DNA molecule continues to unwind and separate, and then it comes back together and closes and rewinds behind it. It's kind of like two zippers. One is opening and one is closing the molecule, and the RNA is being produced where it's open. And eventually this RNA molecule can fall away, and then it's free to be carried wherever to be used. And in eukaryotic organisms, this would occur in the nucleus, and the RNA would move out of the nucleus into the cytoplasm to be used and read in the cytoplasm. Now, a quick look at this, the way it's labeled. You should recognize whether this is DNA or RNA, and you do it by the presence of this nitrogen space right here, this molecule, uracil. DNA does not contain uracil, but RNA does, so this tells you it's an RNA molecule. Okay, so that's the process of transcription, greatly simplified. I don't talk about the RNA polymerase or any other enzymes, just a very, very basic description of it. You're welcome to take a look at the video on YouTube. It should still be working. So to get from RNA to a polypeptide chain, which is an amino acid chain, we're changing language, so to speak. So this is called translation. We're going from the nitrogenous base sequence to an amino acid sequence. And this happens with the help of three RNA molecules, three different types of RNA molecules, and it occurs at the ribosomes. So these three different types of RNA molecules, there's transfer RNA, there's ribosomal RNA, there's messenger RNA, and there's transfer RNA. The little lowercase letter tells what kind of RNA it is. Ribosomal RNA, along with protein, makes up the ribosome. And the ribosome becomes very important. That's where translation and protein synthesis occurs in the cell. And we've talked about ribosomes earlier in the semester, so I won't go back and talk about those a great deal more. Messenger RNA carries the message for the nitrogenous base sequence, which is the message of life. That's the message for coding information. It's in the sequence of nitrogenous bases. That message is carried to the ribosome, which again is made up of ribosomal RNA and protein. And that ribosome serves as a workbench and helps prime the messenger RNA molecule. As the messenger RNA moves across the ribosome, the codons can be exposed and read. It's every three bases on the RNA molecule. The transfer RNA molecule picks up a specific amino acid. There would be 20 different transfer RNAs. Each one will pick up a specific type of amino acid. It brings it in to the ribosome where the messenger RNA is attached. There are three bases on the transfer RNA molecule that will be exposed that will complement the codon of the messenger RNA, and those three bases are called an anticodon. The transfer RNA docks up or binds with the messenger RNA, bringing its amino acid with it. Another transfer RNA comes in beside it. It binds with the next codon, with its anticodon, and the two amino acids that they're carrying are close together and they're bonded together. And this is how the chain of amino acids lengthens. So showing this, here's the ribosome. It's shown as a large subunit and a small subunit. I'm not so concerned about that. There's the messenger RNA molecule that's moving across the ribosome